Today, we're diving into a cool trick in biology, how to actually reprogram a living cell.
So how do scientists turn these little bacteria into microscopic factories that work for us?
Well, the star of the show is usually E. coli, kind of the lab workhorse of biology.
To give it new instructions, we use something called a plasmid. Think of it like a USB stick for the cell.
This plasmid carries the new gene we want, say for making insulin or resisting an antibiotic. Okay, so how do you get this new instruction manual into the cell? It's not that easy.
Well, there's a problem. Both the cell and the DNA are negatively charged. It's like trying to push two magnets together.
The trick is to make the cells competent. Basically, you convince them to lower their defenses. And the first step is a chemical trick, an ice-cold bath of calcium chloride. Those positive calcium ions jump in and neutralize all that negative charge, acting like a bridge. This whole treatment makes the cell wall kind of porous, creating little openings for the plasmid. Okay, the plasmid is now sitting on the outside. The next step, a sudden heat shock. And we mean fast, from ice to a hot bath for just 45 seconds, then right back on ice. That rapid temperature change creates a pressure difference that just shoves the plasmid right inside.
So that's the process. Make cells competent, heat shock them, let them recover, then plate them out.
But here's the thing, this process is super inefficient. We're talking maybe 1 in 10,000 cells works. So how in the world do you find that one successful cell among billions of failures? You use what's called a selectable marker. It's another gene on the plasmid that gives it a superpower. After the heat shock, you spread all the bacteria on a petri dish that contains an antibiotic. Now, only the bacteria that actually took up our plasmid can survive. Everything else gets wiped out.
But this is science, so we need to measure how well we actually did. And we do that by calculating the transformation efficiency. It's basically a score of our success.
So why does this all matter? Well, this simple technique is the foundation for almost everything in modern biotech. We've basically reprogrammed life. The real question is, what should we build with it next?